I would like to thank the organizers for inviting me to speak at this year's conference. I would also like to declare that I do not have any conflicts of interest. Today I will be speaking about the translational diagnostics of donor-derived cell-free DNA testing, as well as some other assays that my lab at the Vancouver General Hospital in Vancouver, Canada, have been developing. Chronic kidney disease affects 1 in 10 Canadians, with estimated costs of over $2 billion a year. Transplantation is a treatment of choice for those whose kidneys fail. Unfortunately, however, it is often regarded as a replacement therapy rather than a cure, given the overall failure rates of graft over time. Long-term survival after 10 years is poor, with severe graft rejection occurring in over 30% of transplant patients, causing premature graft loss, return to dialysis, and early death. While many factors may jeopardize the transplanted organ, the overwhelming cause of graft loss remains rejection. This results when the recipient immune system recognizes foreign markers, of which the most important are human leukocyte antigens, or HLA genes, on the graft and attack the organ. There are 3 main types of rejection. However, it is antibody-mediated rejection which is of particular concern. Antibody-mediated rejection remains the most serious and destructive form. It may occur early or late in the transplant course, and in Canada, AMR is a major cause of chronic graft attrition, and as there are no proven treatments for AMR, prevention is crucial. In 2017, the Genome Canada Transplant Consortium was established with a focus on managing, monitoring, and minimizing antibody-mediated rejection in renal transplant. With a problem so great, we have built a world-class team of provincial and national level programs that cover all aspects of renal transplantation, with involvement from all major transplant programs in Canada, the National Transplant Registry, and over 22 universities across the country. These institutes include expertise from genomics, proteomics, histocompatibility, epitope biology, organ matching, pharmacology, legal and ethical, and socioeconomic specialists. Team members have been drawn in from diverse multidisciplinary backgrounds, working in outstanding facilities to promote the importance of precision medicine approaches to tackling key challenges in kidney transplantation. The current paradigm for kidney allocation and transplantation will be hopefully transformed by some of the work I'll be presenting today. And our future plans will be to use this as a pilot program and roll out the successes here into all other solid organ transplant programs, hematopoietic stem cell transplantation, and other immune-related disorders. Our approach is entirely unique, and Canada is one of the few countries which can contribute the biological, clinical, and translational data to building a precision medicine immune program by having not one, but multiple integrated national strategies for the management of chronic kidney disease. The program is built around 4 key pillars, the first of which revolves around the reduction of risk. Pillar 1 focuses on better understanding and improving the current paradigm of matching an organ donor with its ideal recipient. Pillar 2 is the largest area of work and focuses on clinical translation of assays for monitoring the immune response to the graft. This may involve either monitoring the immune system of the potential recipient pre-transplant, for example, to identify the level of baseline sensitization or activation state of the recipient immune system, to post-transplant monitoring of the health of the graft once it is in place. Pillars 3 and 4 focus on systems pharmacology and the socioeconomic impact of our findings from Pillar 1 and 2. Today I will be presenting some of our findings from Pillar 1 and 2 with regards to epitope matching and immune monitoring. The HLA genes of the major histocompatibility complex are located on chromosome 6 and are the most polymorphic genes in the human genome, with over 25,000 alleles reported, and more being discovered each year, making it almost impossible to match a donor and a recipient for the best genetic match. Because the HLA proteins are expressed on most nucleated cells and are highly immunogenic, these antigens are the principal graft antigens responsible for rejection. Precise HLA matching between identical twins or siblings results in excellent transplant outcomes with little rejection, and close matching of deceased donors and recipients can also reduce AMR and improve survival. Due to the sheer number of allelic variables, matching on the genetic level is prohibitive. However, within the HLA protein, it is only small portions of the antibody binding region that actually contacts the antibody. These are called epitopes, and you can see them on the diagram colored in red, and there are far fewer epitopes than there are alleles. In order to define the epitopes of our transplant population, it was necessary to have single base resolution of the key exons of all 11 relevant HLA genes. To that end, my lab established the first Canadian next-generation sequencing workflow for HLA genotyping on behalf of the Canadian Collective Initiative. We were one of the first HLA labs to implement clinical next-generation sequencing for transplant programs, and since 2016, we have fully sequenced about 5,000 patients. The technology is now being established in multiple HLA labs across the country. Under the new precision medicine program, my lab has been moving into the long-read technologies, where we have been collaborating with Oxford Nanopore on developing their platform and assays for HLA genotyping in a clinical setting. Within the last few months, we have been successful in validating nearly 100 patient samples using the MinION sequencer and a clinically validated assay for high-accuracy HLA genotyping. Our plan is to finish a full validation and transition the entire BC transplantation program onto the Nanopore platform, and if successful, we will be one of the first labs in the world to use this technology in clinical assessment of transplant patients. As I mentioned earlier, the number of HLA alleles are in the tens of thousands, and allele-level matching is not a possibility. The implementation of sequence-based technologies has allowed us to map the epitopes, which are sets of 3 amino acids of relevance, within the antibody binding region of the HLA protein molecule. By having the right technologies, we've been able to map the epitopes of all the alleles observed in our BC transplant population, and this has allowed us to reduce the level of complexity from thousands of alleles to just hundreds of eplets. Based on the work of one of my PhD students, Jenny Tran, we have been able to demonstrate, using real population-level data, the reduction of complexity. Here is an example of HLA-B, where the BC population has 107 alleles, which is converted to only 26 eplets. Following from this, we carried out a simple simulation of provincial transplant program waitlist sizes to identify if epitope-level matching was feasible. The results suggest that a high degree of compatibility is achievable with most waitlist sizes, with the inflection point at about 300 patients, sufficient for most to obtain a mismatch score of 0. The impact of having the translational diagnostic tools to be able to do this cannot be underestimated. Within 2 years of the program start, we are already at the point where the matching paradigm for organ allocation will be changing to epitope-level matching. This has been a phenomenal rate of advancement. I will now move on to the second pillar. As I had previously outlined, this pillar focuses on translating into the clinic assays and methodologies that we can use to monitor, either pre or post-transplant, the state of the immune system or the state of the graft. We have developed a number of diagnostic tools, and assessment of these using our longitudinal cohort of patients is still underway. However, I will now present some of our more interesting findings, including the use of deep immunophenotyping and machine learning, T-cell repertoire sequencing of donor-specific clones, and of course, donor-derived cell-free DNA. Developing biomarkers to guide the use of biopsy and to pinpoint the immune response is key to successful management of the graft, as well as allowing us insight into how to better individualize treatment and therapeutics. Currently, we have no tools in which to assess the immune competence of the recipient, which would allow us to stratify the amount of immunosuppression they would need post-transplant. We also have limited tools that are able to assess immune response, except for serum antibody detection, which is a terminal stage of the immune response, and by then, therapeutic interventions have very limited impact. Early, better, and easily measurable biomarkers will be key to building the immune profile for individual patients, which is the cornerstone of the personalized approach we are proposing. My lab has been able to show at a deep resolution level the immune cell subcompartments that are present and that become activated post-transplant. Our approach has been to take a blood sample at the time of transplant to establish baseline immune repertoire, serial blood collections post-transplant, and at time of event, for example, if a rejection episode is suspected. Using a highly integrated 6-cell subset panel, including B cells, T cells, monocytes, natural killer cells, and dendritic cells, we have characterized the changes in cell quantity, compartment, and activation status for many of the key immune cells in the periphery. Deep cytometry on this level can be complicated to set up, and my lab has also established a workflow that allows for much more streamlined and standardizable workflow, which is now being validated at our partner site in Edmonton. Multi-site validation will add weight to the utility of this assay as a clinical monitoring tool. Machine learning approaches have been employed to understand the complex cellular signatures observed in the study cohort, with up to 13 markers per panel and 6 cellular panels under different clinical conditions. This study design is perfect for the use of machine learning algorithms that would allow my investigators to tease out, hopefully clinically relevant signatures of immune recognition or activation, signaling to the physician that changes in the recipient immune response may require further investigation or intervention. Another assay we are validating to translate into the clinical diagnostic arena is T-cell repertoire monitoring. We have established T-cell repertoire sequencing to identify clonality and diversity in the T-cell repertoire, and in collaboration with Adaptive Biotechnologies in Seattle, have been able to establish a workflow for the identification of donor-specific clones using mixed lymphocyte cultures to start. Currently, we are using DNA isolated from PBMCs and running deep sequencing on an Illumina MiSeq. However, the plan is to also move this to real-time sequencing on the Oxford Nanopore platform. Our protocol involves a 5-day MLC at the time of or pre-transplant, using donor cells as a stimulator to challenge recipient lymphocytes. Cultures are then DNA extracted with the proliferated, hopefully donor-specific clones, and prepared for TCR beta sequencing. Sequencing the recipient repertoire, both unchallenged and challenged, allows us to gather data on the baseline gene utilization of the recipient T cells and identify expanded T cells post-challenge with donor antigen. The key to this assay is to be able to follow patient repertoire clonality and diversity post-transplant. And given that we will have already gathered the donor-specific clone information pre-transplant from the MLC, we would now have a biomarker with which to monitor the possible expansion of donor-specific T cells in patient peripheral blood. Here I show some preliminary data using controls that were stimulated with a disparate MHC control donor. As you can see here, there are 2 distinct clones that appear post-challenge that were not present in the unchallenged repertoire. In principle, this workflow can then now be used for our donor-patient pairs, and we are now moving into validating this as a post-transplant monitoring tool. In addition, we will also begin to sequence the B-cell receptor gene utilization and correlate this with HLA matchmaker B-cell epitopes. The clinical applications of cell-free DNA, either donor-derived in the context of transplantation or tumor-derived in the context of cancer, have been thoroughly reviewed. Our approach to donor-derived cell-free DNA has been to assess its clinical utility, not necessarily as a liquid biopsy replacement, but more of a marker of rejection or graft health. Being able to take multiple samples over time with minimal invasion will provide a clearer understanding of what is happening within, and in conjunction with other clinical markers, for example, donor-specific serum antibody detection or a rise in creatinine, will guide the physician's decision as to when to take the more invasive route of biopsy for final confirmation. The use of a tool of surveillance of health that would allow us to intervene earlier is doubly useful, as it will also impact the problem of organ supply. If we are able to extend the life of an organ, many more people would be able to benefit from a new transplant, and the general organ shortage would be lessened. We have reviewed the utility of donor-derived cell-free DNA in detection of different types of allograft injury in a recent publication in Nature Reviews Nephrology, assessing the different methods of determining donor-derived cell-free DNA, its analytical validity and practicality as a clinical assay, and its diagnostic performance against different types of rejection, namely T-cell mediated and antibody-mediated rejection. It must be noted that elevated donor-derived cell-free DNA levels are not rejection-specific, but more reveals the degree of injury being experienced by the graft. On the next slides, I will present some of the data from my collaborators, for which studies were specifically designed with rejection discrimination in mind, and patients' cohorts were selected as such. Two of our collaborators, Dr. Michael Ulrich and Dr. Eki Schutz, have published extensively on the clinical utility of donor-derived cell-free DNA as a marker of graft rejection. In this 2019 single-center study using droplet digital PCR, about 200 patients with stable versus T-cell mediated rejection, interstitial fibrosis, or acute tubular necrosis were assessed using plasma cell-free DNA collected over the first-year course post-transplant. Not only was the assay able to discriminate between the different types of rejection, the high negative predictive value of donor-derived cell-free DNA, especially for biopsy-proven rejection, would have avoided a number of triggered biopsy, triggered due to the elevated plasma creatinine. This alone is valuable for limiting the increased risk in patients with high biopsy risk. The level of donor-derived cell-free DNA was also assessed as a detection method of inadequate immunosuppression, in conjunction with drug monitoring, often by serum trough levels. The combined values may be helpful in achieving personalized immunosuppression. Donor-derived cell-free DNA has also been evaluated for its ability to discriminate rejection or monitor graft injury in non-renal tissue. Dr. Schutz recently published on the clinical utility of donor-derived cell-free DNA in liver transplantation and showed that this biomarker was capable of earlier and more sensitive discrimination of acute rejection episodes when compared to conventional liver function tests. Other organ assessments have also shown similar discriminatory performance characteristics, for example in heart allograft rejection. We've also developed other uses for donor-derived cell-free DNA, namely as a monitoring tool in therapeutic interventions. In this study, my lab, in collaboration with Dr. Chris Blosser at the University of Washington, have used donor-derived and tumor-derived cell-free DNA to test kidney transplant recipients undergoing cancer treatment with immune checkpoint inhibitors to minimize risk of allograft rejection and cancer disease progression. Kidney transplant patients require immunosuppression to minimize the risk of allograft rejection. However, this then opens them up to higher rates of cancer, especially cancers associated with oncogenic viruses. Cancer therapies have markedly improved in the last decade, with immune checkpoint inhibitors offering better outcomes, with fewer side effects compared with prior chemotherapies. We propose the combined monitoring approach of using donor-derived and tumor-derived cell-free DNA monitoring in kidney transplant patients who undergo checkpoint inhibitor treatment for solid neoplasm, thus allowing us to earlier detect cancer recurrence and progression, and decrease the incidence of allograft rejection, failure, and death, compared to contemporary cohorts without cell-free DNA surveillance. Dr. Michael Ulrich, who gave a wonderful talk yesterday on the value proposition of donor-derived cell-free DNA in organ transplantation, has been collaborating with us for many years, and we have together published on the clinical utility and value of using donor-derived cell-free DNA as a marker of graft injury. The key message is that the main value of cell-free DNA is as a guidance tool for decision-making. Biopsies would likely, at least for now, still be used, but donor-derived cell-free DNA would add to the panel of indicators of allograft damage, such as serum creatinine, estimated EGFR, and proteinuria. Markers of immune activation, for example, donor-specific antibodies, will also be used to assess graft health and identify earlier the need to intervene. And as part of an overall profile, a suite of assays which would be developed into a polyomic risk score based on companion diagnostics for a precision medicine approach is the way that we would individualize management and treatment. And with that, I would like to finish my talk by highlighting the importance of companion diagnostics for effective translational diagnostics. I would like to thank my team in Vancouver, as well as my many collaborators across Canada and internationally.